Hello and welcome to my video about biological molecules. There are 5 types that I'm going to talk about today. First, carbohydrates, then proteins, then fats, then DNA, and then finishing off with ATP. Okay, now moving on to carbohydrate, they are found in foods such as bread, fruit, like banana and orange, yogurt, milk, starchy vegetables, and more. They are stored as glycogen in the liver and starch in plants. Main types of carbohydrate: monosaccharide, disaccharide, and polysaccharide. Monosaccharide is one simple sugar, is the building block of all carbohydrates, and it can be broken into anything simpler. Disaccharide is two monosaccharides joined by a glycosidic bond. A polysaccharide is a polymeric carbohydrate molecule of long chains composed of monosaccharides. An example of a monosaccharide is glucose, fructose. For disaccharide is maltose and sucrose, and for polysaccharide is starch and glycogen. It has two monomers, which are alpha glucose and beta glucose. They can be remembered easily as remembering where the OH groups in these molecules are. The OH groups in alpha glucose are in a down, up, down, down position as in the pictures shown, so you can remember it as that. And you can remember the beta glucose from its OH position as well, which is down, up, or do. Now on to testing for carbohydrates. We put carbohydrate solution and Benedict solution into a boiling tube and then boil the tube in a water bath at about 80 degrees for 5 minutes. And if there is carbohydrate present, then the solution will change color from blue to red. Now, if you test for disaccharide, you would need to add hydrochloric acid into it first and then neutralize it with sodium hydrogen carbonate. And then make sure that the pH is neutral by adding an indicator paper and repeat the test like normal. As you can see, there are 4 main types of polysaccharide: cellulose, amylose, amylopectin, and glycogen. Cellulose, amylose, and amylopectin are all found in plants. The only difference is that cellulose is made of a subunit of beta glucose while the rest is made of alpha glucose, and that amylopectin has a 1,4 and 1,6 bond, and it is branched while the rest has only 1,4 bond and is not branched. And then glycogen is the only one found in animal, and its subunit is alpha glucose and is branched. Lipids are composed of carbon, hydrogen, and oxygen in a long hydrocarbon chain. They are an energy storage molecule with 3 diverse groups: fats, phospholipids, and steroids. They do not form polymers, and their building blocks are fatty acids and glycerol. Fatty acids and glycerol makes up triglycerides, the main lipids in our diets, joined by ester bond between the glycerol and fatty acids in a condensation or hydrolysis reaction, same as carbohydrates. There are also saturated fatty acid and unsaturated fatty acid. Unsaturated fatty acids are more healthy than saturated, and they're often liquids, and they're mostly from plants and contain a carbon double bond, while saturated is unhealthy, often solid, mostly from animal products, and has no carbon double bond. Moving on to phospholipids, they are a molecule which makes up the cell membrane with a phosphate head, which is hydrophilic, which means likes water, and a hydrophobic tail, which makes it arrangement having its head facing the water and its lipid tail facing away from the water. To test for fats, you squash up the food sample and then add it to ethanol. Leave it to dissolve and then add it to water, and if there is fat, the water will have a cloudy white precipitate in it. Okay, moving on to proteins. Proteins are in foods such as milk, egg, chicken breast, meat, and many more. Proteins have a structure of having an amine group, which is the NH2 group, and a side group, which is an R group, attached to the carbon, and then a carboxyl group also attached to the carbon at the side, as shown in the picture here. Okay, moving on to the 4 types of protein. The primary protein is a sequence of amino acids held by peptide bond. Peptide bond is formed by having H2O removed, which forms a bond between the COO and NH, as shown in the diagram above. This is called a condensation reaction, and to reverse this, just add water, which is known as the hydrolysis reaction. Next up, we have the secondary protein, which is when the primary structure twists or folds itself into a 3D shape. An example of this is an alpha helix and a beta sheet. They are both maintained by weak hydrogen bonds. Moving on to tertiary protein is when a polypeptide chain folds itself into a complex 3D structure. Different bonds maintain the shape, such as the disulfide bond, which is a covalent bond and is very strong, and an ionic bond, and a hydrogen bond. Lastly, in the protein section, we have the quaternary structure, which is more than one polypeptide chains joining together, and has a prosthetic group joining onto the polypeptide. And there are 2 types of this, which are globular protein and fibrous protein. Globular protein is round, and an example of this is hemoglobin. And the fibrous protein is a long chain. An example of this is collagen. Okay, moving on to the penultimate biological molecule, which is DNA. DNA is a polymer and is made of nucleotide monomer. The nucleotide monomer has, as you can see from the picture, a phosphate group at the left and a deoxyribose sugar, which means it only has 5 carbon in the middle and attached to a nitrogenous base, which in this case is a purine. Okay, moving on to the purines. The purines are the adenine base and the guanine base. Adenine can form 2 hydrogen bonds, while the guanine can form 3 hydrogen bonds. And then there's pyrimidine, which is uracil, thymine, and cytosine. Cytosine is the only one that can form 3 hydrogen bonds out of these. The others, thymine and uracil, can only form 2 hydrogen bonds. For the base to pair up with hydrogen bonds between it, it has to be between a pyrimidine and a purine. And an exception is that adenine cannot pair with cytosine, even though they are purine and pyrimidine. Lastly, DNA is joined up by a phosphodiester bond between the 3rd sugar and the 5th sugar, connected by a phosphate group. The last biological molecule in this video is the ATP, which stands for adenosine triphosphate. ATP is a universal energy storage molecule that stores and releases chemical energy. It is made up of 3 phosphates attached to each other and to a ribose sugar, and also to an adenine. To release energy, ATP breaks up one of its phosphate group, forming ADP, which stands for adenosine diphosphate, and a phosphate group. This is a reversible reaction, so it can happen. That's it, that's the end. Thanks for watching. Sorry for any mistakes or my bad pronunciation. Leave a like, subscribe, and thank you.